Welcome. My name is Matthias Becker and I'm a team leader for memory-driven computing at the German Center for Neurodegenerative Diseases. Today, I want to present you our work on scaling genomics data processing with memory-driven computing to accelerate computational biology. This is a joint work between the DZNE, the West German Genome Center, the University of Bonn, Hewlett Packard Enterprise and Hewlett Packard Labs. Biological research is becoming more and more data-driven. Microscopes generate high-resolution imagery or movies, and genomic data can be digitalized using the technology of sequencing. The price for sequencing a human genome has dropped well below $1,000, and this technique is used more and more. Datasets can easily go into hundreds of gigabytes, and many of them are being produced today. We are seeing a real data avalanche that is a challenge for the computational processing of that data. If we look at the US National Center for Bioinformatics, they provide a central repository for genomics data that is commonly used by the community. We've queried the database for the number of publications per year, and you can see an exponential growth. Please note that for 2019, not all datasets are available yet. These datasets or studies are only being made available online once the corresponding publication has been accepted. Therefore, there is some delay and the number is expected to grow over the next 12 months. To be able to deal with this avalanche in data, we have been exploring the use of memory-driven computing, a concept that was developed by Hewlett Packard Enterprise. Basically, it is the idea to switch away from the traditional von Neumann architecture in computer design and go for a different approach that actually takes the memory and therefore the data that you actually are interested in as a central component of your system. This is rather than the CPU or the processing component that typically is considered the main part. In the traditional von Neumann architecture, you can see memory is attached directly to the CPU, and from the CPU you can go either into I/O or to the network. With MDC or memory-driven computing, it is the goal to overhaul this architecture and go for a new approach. Persistent storage, like traditional hard drives and DRAM, are becoming one in a persistent storage pool. This could be realized, for example, using the memristor. It is also decoupled from the CPU so that you can have direct network access. Finally, since we're dealing with larger amounts of data, the cost, the energy consumption for transferring that data is becoming prohibitively large. So we produce a lot of, or we consume a lot of energy just for moving that data. So the traditional copper wires will be exchanged into an optics fabric. The underlying idea is that the components are connected using a fabric. An example for that could be Gen Z. So therefore, we will get fabric-attached memory that is available to all the components within the fabric. It is byte addressable, and the Gen Z specification allows for up to 60 million devices that allows us to address 4096 yottabytes. So there's room for scaling. We can connect different device types, so all kinds of processors, architecture independent, also different kinds of storage, so traditional storage, persistent memory, DRAM, and everything in between that we consider necessary. It is also possible to connect accelerators. Typical examples are GPUs, FPGAs, but in principle the interface is also compatible with future approaches, so that you can think of integrating quantum devices once they become widely available. Furthermore, since there's a well-defined interface, it is also possible to connect other devices. In our example here, we have put a sequencer that one day will be able to directly write the data that it produces into the fabric attached memory so that it is available for further processing in the next steps. So when we work with sequencing data, this usually means that the sequencer looks at short fragments of genomic sequences that need to be processed. So the sequencer produces a large number of those, hundreds of millions per experiment, and these short fragments then need to be aligned to a reference genome and can then be processed furthermore. In a classical approach that is commonly used in biology, we have workflows that take the data from disk-based storage, processes it, and writes it back onto the disk. And then we have a sequential runs of tools that all communicate through intermediate files. However, we notice that with this approach, a lot of time is spent on actually reading and writing the data. And therefore, we propose with memory-driven computing to actually keep the data in memory and have the tools work on the data in memory. So this not only allows us to reduce the I/O between the individual steps, but also to run multiple tools in parallel. So we could run an alignment process and a quality control process at the same time, because we don't overwhelm the disk-based storage that was used previously. So since we only operate in memory, we can achieve faster processing speeds. One of the commonly used tools for processing genomic data after the alignment are the Samtools. Samtools have been published in 2009 in bioinformatics, and they introduced the sequence alignment map format and the corresponding tools to actually work with them. They provide a large number of features, but we're going to concentrate on 4 of the most commonly used in this work. First of all, there's a view command. It basically allows you to filter your sequencing data. So you can go for a specific chromosome, or you can filter for a specific quality score, but you can also convert between different file formats. Secondly, you can sort the reads. So remember, you have hundreds of millions of these reads, and you can either sort them by the name that was given during the sequencing process, but you can also sort them by the genomic position. Both approaches are required for different steps. Reads can also be paired, so you sequence the read from different ends, and then you get paired reads. So you have two read informations in your file that belong together. And the fixmate functionality basically gives you a score about the quality of the pairing. And finally, you will be dealing with a lot of duplicates. This is just part of the process or of the processing of the biological samples that duplicates will be introduced. And it is important to mark these duplicates to remove biases or undesired shifts from your experiment, from your data. So we're going to focus on these 4 functionalities. If we now look at the implementation side, so what did we actually do? We took Samtools in its original form and modified it to apply memory-driven computing principles. First of all, you need to know that Samtools can deal with 3 different file formats, SAM, BAM, and there's another one, CRAN. However, this is not used very often, therefore we focus on the SAM format and the BAM format. The SAM format is a text-based format you can see in the upper right corner. It contains some header information, this includes sorting information and some sequence information, and then you have the actual reads. So here we only have one read. It is based on the first chromosome and it starts at genomic position 497. We then can see in the center the actual sequence that it contains and the corresponding quality information. Finally, we have some text like XT, NM, and so on. Another tag that could be included in there would be a tag called MS, the mate score, that would be calculated during the fix mate step. The other format that can be used is the so-called BUM format. It's a binary version of the SUM format. It is both formats are defined in a central format specification, currently at version 1.6. And basically, you get this binary representation. You have a number of blocks that are being, or a number of reads that are being combined into blocks, and these blocks then get zipped and concatenated into one larger file. In our implementation, we now focused mostly on parallel data loading and parallel data saving, and did some small modifications to the processing and in particular the sorting in between as well. Now, we've seen these different commands that were used by Samtools, and they usually are not used isolated. So typically you use a multitude of these commands, and you can do that using different approaches. If you are developing a tool or you need some intermediate results, for example, you could use intermediate files. So you have your input file, then you would sort it by name. You get a next intermediate file that is written to disk. It has to be read then by the fixmate command, and so on. What you see here is actually one very commonly used pipeline. It's a pipeline to mark duplicates. So we talked about the mark duplicates command before. However, the prerequisites of it basically dictate that you have to run the pipeline that is presented here. So you sort by name that your mates or your paired reads are next to each other. Then you can run the fixmate command to calculate the mate score. Then you have to sort by genomic position again, and then you can finally mark the duplicates. As you can see here, we have 3 intermediate files, and if you consider that these files are several gigabytes or dozens or sometimes even hundreds of gigabytes in size, this is quite a lot of overhead. And remember, if you go for the binary format, you also have the overhead of compressing and decompressing them. So the next step would be actually to use a pipeline. So you can actually use the pipe to concatenate the different commands. This way, your data only has to be read once and written at the end again. However, in between, the exchange between the different commands still happens in the digital or in the binary representation. So the data has to be serialized and then deserialized again. You can skip the compression, but you still have to convert it to pass it between the individual steps. Finally, with memory-driven computing, we load the data into the memory using the native format once, and then we just operate directly on that data. So there's no need to serialize or deserialize it. We just have it in memory and directly operate on it. So for data loading and saving, we have implemented a parallel approach, and this actually works for the BUM files as well as for the SUM files. So the SUM file case is straightforward. The file is the format is line-based, so and text-based and line-based. So one entry consists of one line, so we can just take groups of lines and assign them to a thread for processing. In the case of the BUM files, we have these compressed blocks. So to distribute our work, we first need to scan the file to find out the offsets of the blocks. These are not stored anywhere in the file otherwise. However, since our data is in the fabric-attached memory, the actually scanning of the input file can happen rather fast. Afterwards, we would then again distribute the compressed blocks to a number of threads for processing. Now, when we want to save our data, we have our in-memory data where we have the different alignments or reads and the header information. We put them into the proper format and then we compress them. We do this in place, and therefore the maximal size that we reserved, which is the uncompressed size, is not actually used, but instead we get this continuous disk space of file space, memory space, where these blocks are only partially filled. So in the end, we just remove the gaps, we compact it, and we get our final output, our final file that can then be stored to disk. Now, to evaluate our modifications, we use two different hardware systems. First of all, we use the Dell Blade server with 32 cores and 64 threads. It was a system with a NUMA architecture and 2 sockets. And we had 768 gigabytes of memory, so that we can actually keep our data in memory. As a second system, we used an HPE Superdome Flex with 144 cores, 288 threads, 8 sockets, and a NUMA architecture. And here we had 6 terabytes of memory available. Since MDC is in a developing process, we emulated fabric-attached memory using RAM disks. For the datasets, we collected 10 samples from different studies. These studies and samples had different sizes. So the smallest one just had 400,000 reads, while the largest one had over 50 million reads. We grouped them into 3 groups: small, medium, and large. And we made sure that these data sets are coming from different groups, and we're covering a large range of diseases so that we can avoid any bias in our data. So you can see we have some controls. We have malaria, AML, some kidney transplant, influenza vaccination. So there's really a broad range in these data sets. Our initial results showed disappointing runtime numbers. Therefore, we closely examined for the source of it, and we realized that allocation strategy plays a major role in the performance. We tried multiple approaches. So first of all, the default libc allocator. We tried the external libraries jemalloc and tcmalloc. And finally, we wrote a custom solution that actually allocated large amounts of memory per thread and then just assigned them without proper tracking. So this was just to see, to have a baseline of the maximum possible value. However, this can only be used for testing. Our results show that libc is not very well optimized for this scenario, and it always shows the worst performance. We can also see that in most cases, the jemalloc and tcmalloc show similar performance, although in some scenarios, in particular for the BAM files, jemalloc is slightly faster. Our custom solution was mostly the fastest one, but then again, this was just to have a baseline of the maximum possible speed, since we cannot restore any or free any memory again. Then we went ahead and evaluated the single functionality. So we evaluated the view command on the large set of BAM files, the fixmate command, and the name sorting. Now, the results are mixed. We have seen some acceleration in the MDC version of Samtools that show us an acceleration for the view command. However, we've also seen that the fixmate command, for example, is slightly slower. This probably stems from the serial nature of that command, but further investigation would be needed. Finally, we can see that for smaller number of threads, the MDC version of sorting is faster than the original Samtools. However, if you just increase the number of threads at some point, there's an equilibrium reached. In all these cases, we tested on the Superdome Flex for 288 and 144 threads, and the rest of the test was performed on the Blade servers. Next, we look at the pipeline for marking duplicates. And the first bar that you see is the runtime of the individual commands using intermediate files that we discussed. Then we have the results for running the tools with pipes. And finally, we have the runtime for the MDC version. We can see here that, first of all, the fixmate functionality takes up most of the time. We can also see that pipelines or the use of pipes always accelerates the process. And we can see that MDC is faster than using pipes. We can also see that on the Superdome Flex system, we have reached the maximum acceleration, and basically after 64 threads, we can see no more acceleration from adding more threads. So what we have seen in our result data, basically, first of all, Samtools are already optimized for single commands. They have implemented a streaming approach where one thread is responsible for decoding and providing the data, and then another one is actually used to store the data again. We have also seen that the fixmate functionality consumes a significant amount of time, and so far it is not parallelized. Next, we have found that pipelines that combine multiple commands are limited by the data exchange between the individual commands. We have seen that the use of a pipe instead of the intermediate files provides us with an acceleration. However, we also still observe the overhead from serialization and deserialization. With MDC, we have removed the I/O from for the intermediate files. And this basically was possible because we share the data in its native data times, and we have shared memory for data exchange between multiple tools. Finally, we found that the memory allocation strategy is crucial for parsing genomic data. A single read requires at least 2 allocations. So the synchronization overhead for multiple threads is going to become an overhead. And since most of the objects are already pre-known in size, we can work with techniques like bulk allocation. So we want to expand our MDC optimized version of Samtools to cover more of the functionality in the future. And we also want to improve the data exchange between tools by using a common or by defining a common way of memory-based data exchange. Furthermore, we see that it's needed to expand into the analysis part because large-scale single-cell datasets from consortia like the Human Cell Atlas basically cause performance to become an issue for the analysis tools. And finally, we want to work on the integration of multiple data sources, but this increases the computational needs. There are multi-omics datasets where techniques like genomics, lipidomics, and proteomics are being combined. There are also spatial approaches, so where you basically bring together omics data and spatial or image information. And finally, population studies are generating growing amounts of data that are an increasing challenge for the community. Thank you for your interest. You can see the whole team behind this work here. We were funded by the Sparse-to-Big project from the Helmholtz Society, the Cluster of Excellence Immunosensation 2, and the Fast Genomics project. I would also like to acknowledge the colleagues from Hewlett Packard Enterprise, Hewlett Packard Labs, and the Limes Institute from the University of Bonn that really helped a lot with this work. Finally, if you have any questions or feedback, you can contact me at the email address displayed here. Thank you.